And I want to thank everyone for being here to join us today. My name is Sabrina Sholts, and I'm a biological anthropologist and a curator at the Smithsonian's National Museum of Natural History. I'm also the curator of the exhibit Outbreak: Epidemics in a Connected World. I'm joining you all today from my home in Washington, D.C., where I have been working for months. On behalf of the museum, it's my pleasure to welcome you to today's program, Ensuring Vaccine Safety. This is the second program of the series, Vaccines in a Time of COVID-19. The series will continue each Tuesday at 12:30 p.m. Eastern Time for the next 2 weeks. So we've got 2 more left, and I hope that you can join us for those remaining webinars. For more than 2 years, the Outbreak exhibit and its associated programming has been educating the public about viruses that spill over from other animals, pandemic threats, and One Health. I take every opportunity that I can to thank the many organizations and individuals who have supported the Outbreak exhibit, and particularly those that have provided us with funding. In today's program, we will hear from 2 experts who are very familiar with the systems that are in place to monitor the safety of vaccines. But before we begin, I'd like to acknowledge the current events and let you know that the National Museum of Natural History stands with the Secretary of the Smithsonian, Dr. Lonnie Bunch, in expressing our deepest sympathy to the families and communities that have been impacted by discrimination and violence. We'll be sharing Secretary Bunch's response to the events, as well as a resource developed by our colleagues at the Smithsonian's National Museum of African American History and Culture. Talking About Race is designed to provide tools and guidance to inspire conversation about race and to empower your journey. We'll share those links with you in the Q&A. Note that the Q&A box is where we'll be communicating with you throughout this program. We'll post other links there as they come up in discussion, and that's where you can submit your questions for our speakers. So please take a moment to locate the Q&A box, which should be either at the top or the bottom of your screen. That's where you'll type your questions, and we'll do our best to answer as many of them as we can. So now let's get started. I would like to introduce you to our first speaker, Dr. Walt Orenstein. Hello, Walt.
Hi.
Dr. Orenstein is currently a professor of medicine, epidemiology, global health, and pediatrics at Emory University, as well as the associate director of the Emory Vaccine Center and the director of the Emory Program on Vaccine Policy and Development. Some of his previous roles include serving as deputy director for the immunization programs at the Bill and Melinda Gates Foundation, where his primary focus was on polio eradication, measles control, and improving routine immunization programs. Dr. Orenstein worked for 26 years in the immunization program at the CDC, and he served as the director of the U.S. immunization program from 1988 to 2004. We're so fortunate to have him with us today. Welcome, Walt, and please take it away.
Thank you very much, Sabrina. My goal today is to familiarize you with how we monitor vaccine safety, both before licensure and after licensure. Sorry. I have no relevant financial relationships to disclose. First, for pre-licensure activities. Because a vaccine is in a test tube today doesn't mean it's in the arms tomorrow. There is an extensive process that is used by the Food and Drug Administration before it gives licensure to a given vaccine. There are 3 phases of clinical trials. The first phase, phase 1, is usually a small number of people. And the big questions are to determine, is the vaccine safe? Does it seem to generate an immune response that we think would be protective? And are there any serious side effects? It's small. There may be a placebo or a comparator vaccine arm in it. And this paves the way for moving to the second phase. The second phase involves at least several hundred volunteers. Sometimes they're larger. And they look for the most common short-term effects. And this is where they compare the incidence of given illnesses in vaccinees versus people who either receive a placebo or a comparator vaccine. They also measure immune responses as well during this period. If it looks promising, that the immune response looks protective and the vaccines appear to be safe, then the ultimate trial before licensure is called a phase 3 trial. This can be as small as hundreds, but often in the thousands of volunteers. For example, we've heard one of the NIH trials is considering 30,000 people in their study, 10,000 placebo recipients and comparator groups, and 20,000 vaccinees. And this is very important because it helps us in determining whether vaccine is safe. And the way we do that, again, is to look whether given clinical illnesses are more common than in the vaccinees than non-vaccinees. That would be supportive of causation. If they're the same incidence, that would be supportive of a coincidental effect. And then they also look at whether the vaccine is effective. Now, vaccine safety monitoring does not end with the pre-licensure efforts. There is a continuing effort to monitor vaccines afterwards. One, because while we may have used it in tens of thousands in the pre-licensure, it may be going to hundreds of millions. And so rare adverse events may not be found. Number two is there may be risk factors or adverse events that could be made as contraindications for vaccines that may have not been discovered in the preclinical trial. And so that's why post-licensure monitoring is very, very important. And there are a number of systems that are used. This is the first is the Vaccine Adverse Event Reporting System, or VAERS, which allows anyone to report an event, be it a parent, being a vaccine recipient, being a healthcare provider, or what have you. And as I'll show you later, it serves not so much for causation evaluation, but to set signals up for what needs to be looked at more closely. The Vaccine Safety Data Link and the post-licensure rapid immunization safety monitoring are very important in assessing safety, because what they allow you to do is look at the incidence of a given adverse event in vaccinees versus non-vaccinees to help in causation assessment. And the third big group is the Clinical Immunization Safety Assessment Project, which includes 7 medical research centers. And what they do is special studies, such as looking for risk groups for adverse events that may become contraindications or precautions to use of the vaccine. At the CDC, the vaccine safety is coordinated through the Immunization Safety Office, which focuses particularly on VAERS, Vaccine Adverse Event Reporting System, the Vaccine Safety Data Link, and the Clinical Immunization Safety Assessment Network. So what are some of the characteristics of this? Again, VAERS is the frontline system. It allows everybody, for example, I had hives after my second dose of shingles vaccine. I reported that to VAERS, and I did that as a patient, not as a physician. The Vaccine Safety Data Link is the 8 integrated system, health systems, more than 10 million members, and as I said before, allows you to calculate the incidence of the adverse event in vaccinees versus non-vaccinees. The Clinical Immunization Assessment Project has 7 centers, works closely with CDC in setting up studies to look at, for example, risk factors for adverse events. And then the Post-Licensure Rapid Immunization Program Monitoring, or PRISM, is run by FDA. It has the medical records of 170 million individuals, again, to try and look at what is and what is not causally related. And if causally related, what are the risk factors for it? As I said, the VAERS system is the lead for setting up what's called hypotheses. And anybody, as I said earlier, can report. You can find out about it at their website, www.vaers.hhs.gov. And basically what's reported here is a clinical syndrome, a vaccine dose, a lot number of the dose, the date of onset of the syndrome, the date of vaccination, and some other information that helps in setting the process going as to whether this might basically all vaccines we use are qualified to get adverse events get reported to them. It accepts reports from anyone. It rapidly detects safety signals, and it can detect rare things that didn't happen in the trial because we're now dealing with the millions and potentially hundreds of millions. And the data are available to the public. Limitations are reporting people may not fill out the form appropriately or well. The data quality may vary. Some people may be good reporters, others not. There is no comparison group. We know what the incidence is in vaccinees of a given event based on VAERS, but we don't know a comparison group to look at causation. So generally, it cannot assess causality, but it's a great hypothesis generating system, identifying concerns and providing initial safety. And where it came in handy was when I was at the CDC, the first rotavirus vaccine, which was called the rhesus rotavirus vaccine, was associated with a bowel obstruction. And the way we found that out is through the VAERS system. We saw an unusual number of reports, and then we did more sophisticated science studies to show, in fact, that it was causally related. The Vaccine Safety Data Link has a system for active surveillance where we had vaccination records, we have patient characteristics, and we have all their healthcare visits in these HMO databases that again allows us to calculate the incidence of an adverse event in vaccinees versus non-vaccinees. And also it enables us to look at whether there are characteristics of the vaccinees that make them different. And so to control for that and say, hey, having this characteristic makes you more at risk for having this adverse event. And I'd like to conclude with some of the websites that are available for those who are interested. There's the CDC Vaccine Safety website, the VAERS website that I talked about already. The FDA has a website. The National Vaccine Program Office has a website. And then the group that makes recommendations for use of vaccines that are used most that is the main recommending body in the United States is the Advisory Committee on Immunization Practices. They deal with a lot of issues of safety. The National Academy of Medicine is often asked as an impartial third-party group to evaluate safety concerns, and this is their website. And then we have a vaccine injury compensation program, and they have information here. And there are other groups that are really good. Paul may talk about the website that the Children's Hospital of Philadelphia has on vaccine safety, which is outstanding. The National Foundation for Infectious Diseases, NFID.org, has a great website for safety issues, and vaccinateyourfamily.org has also a great website discussing vaccine safety issues. Thank you very much.
Thank you so much, Walt. That was fascinating. That was useful. Those links there at the end in particular are great resources for people to go to find trusted, authoritative information to learn more for themselves. Wonderful. Okay. So now we are actually going to go to our second speaker. We've been so fortunate to have two experts with us here today. And then following Dr. Paul Offit's presentation, we will start to get into the question and answer section of our presentation. So Dr. Paul Offit is the director of the Vaccine Education Center and professor of pediatrics in the Division of Infectious Diseases at the Children's Hospital of Philadelphia. He is also the Maurice R. Hilleman Professor of Vaccinology at the Perelman School of Medicine at the University of Pennsylvania. Dr. Offit is an internationally recognized expert in the fields of virology and immunology and was a member of the Advisory Committee on Immunization Practices to the CDC. He's a founding advisory board member of the Autism Science Foundation and the Foundation for Vaccine Research. Dr. Offit is also the author of 7 medical narratives which have received numerous awards. And for what it's worth, I've read several of them and personally recommend that you all check them out. So thank you, Paul, for being with us today, and please take it away.
Just get this up here. This, that one. Okay, thanks, Sabrina. So I mean, the typical length of time that it takes to make a vaccine is about 15 to 20 years. We currently have a vaccine against SARS-CoV-2, the cause of COVID-19 disease, that we anticipate is probably going to take about 1.5 years. So I think people can reasonably ask the question, are we skipping critical steps, the kind of steps that Dr. Orenstein just alluded to? Are we cutting corners to the extent that this vaccine may be less safe than we would ask from a traditional vaccine? So this is an unprecedented moment in time. Never before have we had more interest across the globe in making a vaccine. There have been 120 investigational new drug applications submitted to the FDA. More than 70 companies across the globe are interested in making this vaccine. Groups like BARDA, which is the Biomedical Advanced Research and Development Authority, has given billions of dollars to 5 different companies, and as has the Gates Foundation and World Health Organization. The good news also is the virus is relatively stable. We know what we're interested in. We're interested in that protein that sits on the surface of the virus. There's one particular region that is stable, and that if we can make an antibody response to that spike protein, we can prevent the virus from binding to cells, which means we can prevent the virus from infecting cells, which means we can prevent disease. So that's all good news. The other piece of good news is that every strategy that has ever been used to make a vaccine is being used to make this one. So some groups, like groups in China, are taking the virus and killing it in the same way we make the inactivated polio vaccine, or the hepatitis A vaccine, or the rabies vaccine. There's a group working with the Serum Institute of India that's using a live attenuated viral vaccine, like the measles, mumps, and rubella vaccines. There's groups like Novavax and Sanofi, which are using a purified protein vaccine, meaning you take that spike protein and then purify it and then give that as a vaccine. It's the same way we make the human papillomavirus vaccine and the hepatitis B vaccine. Then there are vectored vaccines, where you take a virus that is generally harmless and you re-engineer it so that it expresses, in this case, that spike protein that's on the surface of coronavirus, which is similar to the way that we make the current dengue vaccine and the Ebola vaccine. In addition, there are also replication strategies or vaccine strategies that have never been used before. So one is to take a virus like adenovirus, which is one of the causes of the common cold, and then you make it so it can't reproduce itself, but that it can make proteins, certain proteins, in this case, the protein that codes for that, or the protein that sits on the surface of coronavirus, that spike protein. Or you can give messenger RNA or DNA vaccines, where you provide the genetic material and then your body makes that protein, in this case, the spike protein. So all of that is on the table. The way that this is been truncated is in a few ways. One is that invariably one does animal model studies. I mean, you take an animal, an experimental animal like a mouse or a monkey or a guinea pig, you inoculate it with the virus, in this case SARS-CoV-2, and you hope that it gets the symptoms and signs similar to what we see in humans. And then you do so-called proof of concept studies, which is to say you try your vaccine out, then you give the animal the virus to see whether the animal is protected. It also lets you, frankly, literally dissect what part of the immune response is associated with protection. Then one does the dose ranging studies, which are sort of phase 1 studies that Dr. Orenstein stated, where you make sure you're not giving too much of your vaccine or too little of your vaccine. And then the phase 1 and 2 studies are generally been relatively smaller than the phase 1 or 2 studies that you see typically for licensed vaccines. The one thing that can't be skipped is the phase 3 study. And Dr. Orenstein stated this. The NIH group that's been put together by Francis Collins has recommended trials of about 30,000 people, either 20,000 vaccinees and 10,000 placebo recipients, or a 1-to-1 trial where there's 15,000 vaccinees and 15,000 recipients. And then and only then can you really make a statement about efficacy. You don't know how long that your vaccine is going to be, for how long it will be effective, but you know that at least at a certain level, let's say 70%, it's effective. And similarly, with 20,000 vaccinees, at least you can say that the vaccine doesn't have a relatively uncommon side effect. I mean, 20,000 vaccinees is not 20 million vaccinees, so you're not going to know whether there's a rare side effect, but as Dr. Orenstein stated, there are systems like the Vaccine Safety Data Link or the PRISM system, which are able to pick up rare adverse events post-licensure. Typically, the FDA will license a product. I mean, and usually it takes about 10 months to license a product. If it's an expedited review, it takes about 6 months. None of that's going to be true here. These aren't going to be licensed products. They're going to be all approved under the emergency use authorization, which is frankly a lighter touch by the FDA. It's not going to go through the typical FDA Vaccine Advisory Committee, which has happened frankly for every other vaccine in this country since the 1930s. The one thing that I want to caution against, and I'm optimistic this won't happen, but just have to be careful that it is an election year, and you want to make sure that we don't skip the phase 3 trial, that we don't just sort of reach our hand into the warp speed bucket. What warp speed is, and I didn't mention this, is it's a manufacturing thing. So what the government has decided to do is to basically mass produce these vaccines, the 5 vaccines that are currently under consideration in the United States, make millions of doses, make tens of millions of doses, even though we don't know whether the vaccine is safe yet, even though we don't know whether the vaccines are effective yet. But when they're shown to be safe and effective in a phase 3 trial, then we're ready to release them to the American public immediately. This is not the first time we've done this. We did this in 1976 for a swine flu vaccine, and we did it in 1955 for polio vaccines. There were 5 companies that also made the polio vaccine at risk, meaning not knowing whether in a large trial it would be shown to be safe and effective. And when it was, then the vaccine was immediately available. You want to make sure you don't skip those phase 3 trials, because that would be a disservice, frankly, to the American public. But I'll stop right there and thank you for your attention.
Thank you so much. Fantastic. I have so many questions. I imagine our audience does as well. For those of you watching in our audience, please enter your questions as they come to you. If you want to direct them to Dr. Orenstein or Dr. Offit in particular, you can write that into the text box. And we'll be taking those after I get to ask a few things that have come to me with both of your presentations. So firstly, and it's really picking up on where we left off with Dr. Offit's presentation, I believe, but it was addressed in both of them. This question about skipping a trial, skipping a phase, like phase 3 is probably said again and again, that's really important. That would be a disservice, to the public to do that. I've just learned by reading the news that China has approved a vaccine, I guess, for military use, skipping, I think that final phase. And that goes to the more broader question, I guess, of are there different standards for vaccine safety in different countries?
Right, I'll start out off and then turn it over to Walt. I would say the answer is generally not. I mean, as a general rule, all countries sort of follow that phase 1, phase 2, phase 3 rule, and then have post-marketing surveillance systems to look to make sure that there isn't adverse events afterwards. It was surprising, I think, that China vaccinated their military. The strategy that the vaccine they have is to take a replication defective adenovirus type 5. So again, type 5 is a fairly common adenovirus that circulates, to make it so that it can't reproduce itself, can't cause disease in people, but can still express that protein, the spike protein, which will then hopefully induce antibodies that will prevent virus cell binding. They did publish, to their credit, the group that's making this did publish a paper, frankly, one of the few papers that's been published about vaccines. So you could look at to what extent the vaccine was safe, and certainly at a higher dose, it did have a safety issue, and look at to what extent it consistently induced an immune response, which also is somewhat questionable. So I'm surprised they didn't submit that to a phase 3 trial, but I'll be curious to hear Walt's comments.
Yeah.
I would agree with Paul. First of all, there are regulatory authorities in many countries. Some have regional medical authorities, as the European Medicines Agency, but they generally follow the same rules. as the FDA does in the United States. I think the big issue here, and that we have to be very careful of, is people are seeing all these deaths, all these hospitalizations, and wanting to do something. But we have to be very careful because we, one, need an effective product, but we also need a safe product. And that's why, in my opinion, a phase 3 trial is really critical. But as I said in my talk, it's also critical that we have continuous monitoring, as we do with every other vaccine, as to whether there are other events we didn't pick up in the clinical trials, and if there are risk factors for those events that we could then make contraindications to the vaccine. But the problem I think people are seeing is all of these deaths and hospitalizations and spikes in the cases. And the only thing we have at the moment is really social or physical distancing and isolation, and a limited number of therapeutics, which at this point are not, in my opinion, wonderful therapeutics. So we need to balance that with a vaccine that might prove to be unsafe.
Yeah. That's interesting. We hear a lot, and I think we're learning a lot about how much advancement we've had scientifically in developing vaccines over these decades, as we learned from both of you. There's so many platforms, ways to develop vaccines now. And I imagine, and you can, either one of you can answer this, that we've seen vast improvements on how we monitor safety. I guess now what you're talking about with the post-licensure monitoring, I mean, that would not maybe have been possible or as effective or rapid at a time before we had, digital surveillance and sort of global communications and things like that we see today. Is that true?
Well, exactly. I think, to me, for example, having a vaccine safety data link is very, very helpful to determine the incidence of a given clinical syndrome after vaccination versus not after vaccination. And if it's higher, then that would be compatible with causation. The other thing that it does is it allows you, if it is causal, to determine what the risk is. And then it allows you to balance the benefits and risks, because no vaccine is 100% safe. No vaccine is 100% effective. What is done is to balance the risks and benefits. And for the vast majority of vaccines, all those recommended, the benefits far, 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 far outweighing risks. But I think with the system we have in place, it not only allows us to determine whether vaccine A causes event B, but it also allows us to determine what the incidence of event B is, to balance it with how many, is that a risk worth taking because of all of the problems with the vaccine preventable disease.
Paul, did you want to say anything to that as well about advancements in safety?
No, I think he covered it.
I don't know how even you would be able to do what we can do now with the internet, for people knowing where and how to report. And being able to do that, in a way that allows, a rapid response against to actually.
And that's what you can do with VAERS. When I had my adverse event, I went on the internet and reported it. And it allows people to report, and it makes it easier for the people doing evaluations of it because they have all these things online.
Well, thinking about all the different vaccines that are being developed now, candidates, and the different ways that that's being done, are there any methods or platforms in particular that are most concerning or most in need of a particular attention or focus in terms of safety versus others?
Oh, you probably would.
Yeah, what I would say is that there were two events that occurred in the 1960s that were sobering. One was an RSV, or respiratory syncytial virus vaccine. The way that vaccine was made, it was made actually at NIH, was you take the virus, grow it up, and then inactivate it with a chemical. In this case, the chemical was formaldehyde. What ended up happening when children got that vaccine and then were exposed to the natural virus is you were actually worse off getting the vaccine. You were actually more likely to be hospitalized, more likely to develop pneumonia, and a couple children died of an unusual pneumonia. And I'm going to tell you why I'm telling you this story as it relates to the SARS-CoV-2 vaccine. Also in the 1960s, there was a measles vaccine that was made by taking measles virus and inactivating it. Again, children developed an unusual syndrome when they were then confronted with the natural virus, the so-called atypical measles, which was again associated with pneumonia and hospitalization. Both of those viruses have something in common, which is that the way that they attach to cells is they fuse to cells. Many viruses don't actually fuse to the cell before they enter it. Rather, they just sort of attach and then get taken up by a process called endocytosis. These were fusion proteins. Now, that's also true of this virus. The spike protein of SARS-CoV-2 is a fusion protein. It fuses to cells. And so you wonder that when you inactivate it, you want to make sure that the conformation of that surface protein is not altered from the way it normally looks on the virus. Now, there are groups, actually 4 groups in China that are using an inactivated vaccine. They actually didn't inactivate it with formaldehyde. Rather, they chose something called beta-propiolactone, which is the same chemical that's actually used to inactivate the rabies vaccine. So I don't think it's going to be a problem, but I guess that's one thing that I worry about is when you sort of don't mess around with fusion proteins. As long as they look like they look on the natural virus, then you're good. But if you alter it, you can run into a problem.
I think that's a very important point. Our goal with a vaccine is to induce a protective immune response. The problem at times is the immune response can mutiny and can cause problems. And that's an example of what happened with the killed measles vaccine and the respiratory syncytial virus. And so we need to make sure that doesn't happen with the vaccines we're currently developing. That's why we need these systems in place to try and detect it if it is, and if we could see any risk factors for it that can then lead to contraindications in specific groups, but allow vaccination of the rest of the population.
That mutiny that you referred to, is that what's called enhanced disease?
It's called enhanced disease, and in fact, one of the issues of why COVID-19 is associated with some of the really bad illnesses is an overactive immune response. In fact, some of the therapeutics being considered are not ones that destroy the virus, but modify the immune response to avoid getting the more dangerous immune response. So that's what I'm talking about. So in fact, there is a natural precedent with SARS-CoV-2 of potentially altering the immune response and leading to some of the severe disease we see now.
You both are so good at explaining this, especially the details, the process, the science, and all the technical concerns that so many people want to hear and need to know. And I'm just wondering if, for example, a parent needed to talk to a child or someone, you need to speak to someone who is really concerned about what might happen, to their child, a potential injury or disease as a result of vaccines. Could you give suggestions for how one does that? What kind of language or I guess a phrasing you can use or points you can make to very simply explain some of this to someone?
So I guess I'll start. I think what surprised, one of many surprises that's been offered by this novel coronavirus, this bat coronavirus that's just made its debut in the human population, is that it causes an unusual syndrome in children, so-called Kawasaki-like syndrome, which basically means it's a multi-system disease, multi-organ disease that can also be associated with shock, the so-called toxic shock-like syndrome. Human coronaviruses don't do that. So the fact that this virus did, and frankly, it didn't seem that SARS or MERS, the two preceding novel coronaviruses, did that either. So I think this has come up. I mean, so now knowing that this kind of syndrome can occur in children, what are you doing when you're making a vaccine to try and make sure you avoid that? Well, I think what that all depends on is what's the reason that children get this Kawasaki's-like syndrome? Is it because the virus itself goes into sort of the lining of vessels, blood vessels, and then damages that lining, so-called endothelial cells? Is it an autoimmune response where as you're making an immune response to the virus, you're also making an immune response to yourself? Is it just that when you're infected, you induce this sort of cascade in your immune system of cytokines and chemokines that's then damaging these endothelial cells? And I think until we know that better, we're not really sure how to avoid it. I mean, my suspicion is that it's the last thing that I said. It's just this general sort of chemokine cytokine response for a virus that's reproducing itself thousands of times in your body. So I think that therefore is very unlikely to be mimicked by any of the vaccines that we're giving. You could argue a live attenuated viral vaccine could do that, but again, I'm confident that the vaccine strategies that are currently being used will not cause that kind of reaction.
I agree. I think the major issue is we have new platforms that, as Paul said in his presentation, that we don't have much experience with, particularly the gene-based vaccines, the messenger RNA and the DNA-based. But that's why we're going through the trials with them, to try and detect if there are any problems and to determine how effective they are.
Yeah. I think what I often hear most is that a vaccine in general, that it is far more beneficial, right, than anything that would happen with the disease.
Exactly, and there is a very lengthy process even after licensure with regard to what gets recommended for use, and this is led by the Advisory Committee on Immunization Practices of the ACIP, which works with CDC, and basically their meetings are in public. They go over, they present data. You can go on the CDC website and you can look at the presentations, you can look at the slide sets, et cetera. And they're very important. They come from many disciplines, medicine, epidemiology, vaccinology, pediatrics, economics, and that's what their job is to try and look at all the information that's available and try and determine what's the best way to maximize the benefits and minimize the risks.
Well, we've got a lot of questions coming in now from our audience, so I'm going to go to those if you guys are up for that. Our first is actually directed to you, Walt, and that you said VAERS cannot assess causality and rapidly detect safety signals. To this person, that seems contradictory. Can you address that? That seemingly.
Okay, if I said it can rapidly detect safety signals, causality is not as easy to do, because the way most of the clinical syndromes that we talk about that can follow vaccines can occur in the absence of vaccines. It's not like we're dealing with a completely unique clinical syndrome we've never seen before. With VAERS, all we know is clinical syndrome A followed vaccine A. We don't necessarily know if it's causal or not. That's why we need to go to things like the Vaccine Safety Data Link to look at the incidence of syndrome A in vaccinees and non-vaccinees. The other thing, though, that VAERS can do, and I gave you the example of the original rotavirus vaccine, the one that's no longer available, that caused a bowel obstruction called intussusception, is we saw a cluster of reports of these within a few days of vaccination, which looked very suspicious. And that led us to do the more sophisticated studies and show, in fact, that the incidence of bowel obstruction or intussusception was higher in vaccinees and non-vaccinees, and we detected a risk of roughly 1 in 10,000 immunizations was associated with this bowel obstruction. And we stopped use of that vaccine because of that. And in fact, the next couple of rotavirus vaccines, before they were licensed, they had to do huge clinical trials to try and show that they didn't get involved in the bowel obstruction problem. So all that came from our vaccine monitoring system.
I think just one thing to add to what Walt just said is I think the word signal is confusing to people. When they hear the word safety signal, they assume that means there's a causal relationship between the vaccine and between the safety issue. Really, all it means is that it's something to look into further to see whether that association between one thing and another was a causal or temporal association. But I can see where the word signal can be confusing.
Yeah,
it's very helpful, Paul. Thanks.
Yeah. Okay, well, I got another question for either one of you, both of you. Since there is likely to be more than one vaccine, how does someone decide which vaccine to use? How does one determine when to receive a vaccine after it becomes available?
One thing that the ACIP will often do is tell you whether they consider the vaccines equal or whether one is they consider superior to another. And my feeling would be is if they consider vaccine A superior to vaccine B, I would try to get vaccine A. Or they may say that in some populations it doesn't matter, in other populations vaccine A is better or whatever. But I would follow those recommendations. And the real key is your healthcare provider and to talk with your healthcare provider as to what it is. One of the things that also it's not clear at this stage how many doses will be recommended, whether they will be single dose regimens or multi-dose regimens. And what will happen if you got vaccine A for your first dose, but when you went back, they didn't have vaccine A anymore, they only had vaccine B, can you get that? And that also is usually addressed by the ACIP.
Yeah, the only thing to add is that when you do these trials, you want to make sure you have a representative population that you're about to give it to. So you want to make sure that you have, an adequate representation of African Americans, Hispanic Americans, Asian Americans, the elderly, people who have comorbidities, women, including pregnant women. And so as these vaccines are tested, there may be subgroups that are better represented in one trial versus another, or certainly better served by one vaccine or another. And that's exactly what Walt said. That's what the ACIP's job is. It's to sort out sort of what you know from these data and then to make the best recommendations for specific groups.
Well, we've got a question here from Samuel Liao, who is asking, have there been any failed attempts during a trial resulting in serious side effects or even deaths? I know, Paul, you'd spoken to this a bit, but maybe you'd like to emphasize some points here for those who may not have caught all of that. Just previous examples of anything maybe going wrong in a trial. How common is that? When has that happened in the past?
Sure. Well, most recently with the dengue vaccine, it was found in that trial that some children who'd never been exposed to the virus before, when they were given the vaccine and then exposed to the virus naturally, actually did worse. And you were better off having either been having been naturally infected with the virus before you got this vaccine than not. And that's, that actually ended up causing the deaths of children, about 14 children in the Philippines. That's the most recent example. But yeah. I mean, the history of medical breakthroughs is sort of littered with human tragedy. That's always true. We expect breakthroughs to be for free, but the only way to really learn about things in people is to test them in people. I think we jump with a large safety net in this case. I mean, so if you take this example, as Walt said, if we have 20,000 vaccinees, 10,000 placebo recipients, and you see that it's safe in 20,000 vaccinees, that tells you that it doesn't have an uncommon side effect, and that it's effective at least for a certain period of time at a certain level. Let's say 70% effective at protecting against moderate to severe disease. You're talking about a virus that's killing 1,000 people a day. So the benefits are huge. And although any vaccine, any medical product that has a positive effect can have a negative effect. It's always, it's true in medicine always, is that You want the benefits to clearly and definitively outweigh the risks, but there are no risk-free situations. The choice not to get a vaccine in this case is not a risk-free choice. It's a choice to take a different, and I would argue more serious risk if we can get vaccines that are safe and effective. So that's always the calculation.
Yeah.
And the choice affects not only you, but potentially the community if the vaccine also reduces transmission, because there are people who may not make good immune responses to it, such as those who have compromised immune systems. And when you're vaccinated, you're not only doing it for yourself, but for those people, because you're helping to break the chains of transmission. What keeps things going is a contagious case finds a susceptible, transmits. That susceptible later becomes a contagious case and transmits to someone else. And what happens there is you have a chain of human to human transmission. We don't have infinite contacts. And when you have high levels of immunity in the population, the likelihood that a transmitting case will find the susceptible is markedly reduced. And so you can break the chains of transmission and indirectly protect people, for example, who can't make a good immune response to the vaccine, or people who are very much at risk and for whom vaccine may not at the time be recommended because there is inadequate safety and effectiveness data. So again, it's two ways that vaccines protect for most of the diseases, direct by an active immune response and indirect by breaking these chains of human to human transmission and reducing exposure of remaining susceptibles.
Yeah. We have a question here from David. Will the SARS-CoV-2 vaccines have VAERS and PRISM coverage, even in skipping FDA approval? And maybe here we can address the difference between approval and licensure as well.
For VAERS and for VSD, let's start with VSD. That's based on the medical records in the HMOs that are enrolled in the VSD. So yes, if a vaccine is being given to their population, they will have that data. Similarly, for VAERS, anything can be reported. So events can be reported to VAERS.
Yeah. Okay. Good. Good to know. That's very important. All right. I have a question here from Roz. How will we know how frequently to get vaccinated, short of seeing a reemergence of the virus, say after 6 months from when the vaccine was given? Great question about
the CDC in particular does surveillance and basically will look at who is getting disease and is it the result of failure to vaccinate or vaccine failure. If it's vaccine failure, they will look at risk factors. So is it that it was a long time ago that you got your vaccine dose or a short time? If it's a long time and you compare it with other people who more recently been vaccinated and they're not getting ill, that would suggest waning immunity and the need for a potential booster response. So these things will be tracked, and it's critical that we continue to maintain very high level surveillance for COVID-19, regardless of whether we had a vaccine or not, to look at these issues, looking at which communities are effective, which groups are sort of gender, which race, ethnicity, geography, all of these are critical in order to decide what actions can be taken to minimize the burden.
Okay, great. A question here from Janice. Interesting, I think. Question, since social distancing and masks significantly influence exposure to the virus and risks, as we know, how are those conditions and those behaviors among vaccinees are controlled for in a phase 3 trial? Do you want to take that one?
I think, of course, you want people to protect themselves as much as possible. I'm sure that everyone who will be entering these trials, which are obviously associated with the consent form, will be told to do all the hygienic things that will lessen their risk, which is ironic when you think that the only way that these trials are going to be of value is you have enough disease in your placebo group so you can make a comment about efficacy. So on the one hand, you're telling them, please don't get infected. On the other hand, you're sort of hoping that enough get infected in the placebo group that you can make a comment about or a statement about efficacy. But I mean, yeah, certainly that didn't affect the AIDS trials. And, in the AIDS trials, the same thing. People were told to practice, the kind of safe sex that wouldn't allow them to get AIDS, but that didn't seem to matter. That still happened. So I think, in all likelihood, we won't be able to significantly reduce the risk because I'm sure some will still continue not to wear masks or not to practice social distancing.
Yeah. Okay. Well, I mean, Janice has another question. Some good ones here. Because the virus, SARS-CoV-2, can infect some people without producing any symptoms, so therefore there's nothing to report, how is that controlled for in, for example, a phase 3 trial?
Well, what you'll do in the phase 3 trial is you'll have certain clinical endpoints, which will be, that you prevent, associated with being SARS-CoV-2 positive, that you prevent, coughing or shortness of breath or fever, et cetera. So there'll be those clinical endpoints, but people will be constantly monitored to see whether or not they're acquiring this virus. So you'll know, I think, whether you're preventing asymptomatic infection associated with exposure or mildly symptomatic infection associated with exposure. I think that's a much higher bar. I mean, what you're really trying to do, this is true for any mucosal vaccine, and that's this virus, you want to try and prevent moderate to severe disease. You want to keep people out of the hospital and you want to keep them from dying. That would be a reasonable expectation for this vaccine. And that it should protect people for a few years would be a reasonable expectation for this vaccine. But I think to have the so-called sterilizing immunity that we use, as it's called, sterilizing immunity with, like, as you see with the measles vaccine, where not only do you not get moderate to severe disease, you don't get mild disease, you don't even get reinfected. I mean, those viruses just bounce off you because that's the level of protection you get with that kind of vaccine. That would be an unrealistic expectation here.
Yeah. Have we dealt with a virus that has so much asymptomatic infection? Like SARS-CoV-2 in dealing with
polio had a lot of asymptomatic infection. I mean, roughly only one of 200 people who got polio, infected with polio, got paralyzed. So there was a vast undercurrent of that disease that you never saw.
Exactly. I think and that's part of the monitoring system as well, is to try and look for that. But usually, if in fact there are people who are getting asymptomatic infection and transmitting, you'll see some people who come down with a full-blown illness. And so you need to continue that. It's not like we've seen 100% asymptomatic illness with a few exceptions. You wouldn't expect that. You would expect some people who would eventually develop the full-blown illness.
Okay. Well, there's a question from Patrick here that deals with the concept and a term that we hear a lot. How effective does a vaccine have to be to create herd immunity?
There's again, we don't know that much about this yet, but there have been some estimates of herd immunity. There's a number called R0, which is called the basic reproduction number. And what that means is it's the number of actual secondary cases. If I were a transmitting case living in a 100% susceptible population, who I would transmit to. So for this particular virus, most people have estimated the R0 at about 3. So the herd immunity threshold is R0 minus 1 over R0. So let me explain that. So if you have 67% of the population immune, and I would transmit normally to 3 people, I would transmit to only 1 person because the other 2 would be immune. And level transmission would occur. If the immunity level went above 67%, I on average would transmit to less than 1 person, and transmission would terminate.
Yeah. Did you want to add to
But
that?
it's not to say it's not good to unless you're at herd immunity threshold, the vaccine is worthless. That's
Right.
not the case. The vaccine can be very helpful to vaccinees, but also, while it may not end transmission, it could dampen transmission and therefore indirectly protect a lot of people. So the numbers of cases you see may be small.
Yeah, the only thing I would add is that you know, building on what Walt said, that if you need to vaccinate, say, 2/3 of the population to stop transmission, they're likely to be, with the exception of the live attenuated viral vaccine, these are likely to be 2-dose vaccines. And so you're talking in this country about trying to give more than 200 million people 2 doses of vaccine. That may take a little while, and that assumes everything works perfectly. So I think we're, assuming the virus continues to do what it's doing, it may take, more than a year certainly to get to that point.
Yeah. Well, we've got a little bit of time left, I think, for one more question, and one that I'm happy to take and have you guys address, because I think it's important. Certainly today, when we're learning, for example, about travel restrictions going into the EU, for Americans and things like that, and realizing, what's happening professionally, personally, for so many of us with this global distancing that seems to be the situation right now. How does internationally cooperation and standards play into vaccines being created and tested for wide application? Would vaccines created in other countries be available to us in the US or vice versa?
It depends on the manufacturing capacity and demand, but hopefully that would be the case. I think the good point here is we are a global community. This virus didn't originate in the US, it came to the US. And it's in our self-interest, not only to try and protect ourselves, but also to try and help other countries reduce their burden. It decreases the risk of introducing it here. And it's unlikely we're going to have 100% effective vaccine. So even if we have all the doses we want, if let's say we had a 70% effective vaccine, that will mean 30% of vaccinees would have to be indirectly protected. And the way to do that is to achieve high vaccination in our own country, but also to try to work to get other countries vaccinated to reduce the risk of somebody either traveling there and coming back with it, or vice versa, somebody traveling from there to here. And the reason we're not being allowed in the EU is we have a heck of a lot more virus, and there is more risk to them. They're perceiving more of a risk from travelers coming to the EU or not. We need to work together, particularly once we have a vaccine, to try and achieve high immunity, not only in our own population, but in every population around the globe.
Wow. Thank you.
I couldn't say it better.
Go ahead, Paul. Oh, Paul, go ahead. Sorry.
I'm sorry. Yeah, I may be breaking up, but I certainly couldn't say it better what Walt just said.
Yeah, I agree. And you know what, Paul, you just said what we say in the outbreak exhibit. Working together, is one of our primary messages, in addition to One Health, and thinking about ourselves as part of a global community is so important. So I think that's the perfect way to actually wrap up here. I want to thank you both for your time, your expertise, taking some of these questions and helping so many folks out there understand more about this really important process that we're all, to some extent, in some way a part of. So to our audience, thank you for joining us. We hope that you'll join us next week for our third webinar in this series, From the Laboratory to a Shot in the Arm, where we're going to explore how a vaccine proceeds through the distribution to eventually end up in your local doctor's office. Our speaker will be Linda Stewart, Deputy Director of Vaccines and Human Immunobiology at the Bill and Melinda Gates Foundation. You can register for the webinar on the NMNH events page that will be shared in the Q&A. Please take the survey that will pop up once you leave the Zoom webinar, and you can always rewatch this program on the webpage for it shortly after we end here, it will be posted. So thanks again, everyone. Please be safe, stay healthy, and have a good day. Bye.
Bye-bye.